Okay, in this tutorial we're going to talk quickly about what facilitated diffusion is. So, facilitated diffusion is a type of diffusion. That means it's a type of passive transport that doesn't require energy. So diffusion and osmosis don't require energy. They're passive processes. They rely on the kinetic energy of the molecules themselves. However, in this case, it's facilitated, it's helped by certain proteins in the surface of the membrane. And so we'll have a look at that. So facilitated diffusion is a type of passive transport that uses the aid of transport proteins that are embedded in the cell or plasma membrane. So if we look at this diagram, essentially the reason you need facilitated diffusion is that some molecules, you can see here you've got a higher concentration of molecules, of these square molecules on this side, so they would normally diffuse in this direction, so from an area of higher concentration to an area of lower concentration. So we'd normally get a net movement in this direction, but there's this damn barrier in the way, the membrane, and it's especially this part here, these hydrophobic tails, that often represent a challenge, because often molecules that are dissolved in water are either polar, so they hydrogen bond with water, or are charged, and because of that they don't bond or interact well with the hydrophobic or non-polar tails of these phospholipids, these lipid tails. And the reason for that basically is that the bonds between these molecules, the interaction, intermolecular forces between these molecules and the polar water molecules is stronger than with these, so that the bond, that kind of prevents them entering this layer, because it's there's a stronger force of attraction to the water here than there is to these tails. So now some really small molecules, so carbon dioxide, oxygen, even water, can in small quantities essentially diffuse through the membrane. They're small enough that they can sort of get through there, and it's also temperature dependent, so the temperature will depend how much kinetic energy they have and whether they're likely to be able to get through there. However, larger molecules or molecules that are highly charged or polar or large polar molecules are going to find it especially difficult to transverse that hydrophobic layer. And that's where the help of these proteins come in. And these proteins can be as simple as basically a protein that has a gap in the middle that acts like a tunnel. And so the amino acids on the outside here, in this region here, sort of just make it a bit darker there and here, they will be hydrophobic, non-polar, interact with these hydrophobic tails, whereas the ones lining the inside will most likely be hydrophilic. They may be charged or polar, and essentially provide a hydrophilic or a charged or a polar environment through which these molecules can move down their concentration gradient. So it's just basically a tunnel. So they're sometimes called protein, usually called protein channels, sometimes referred to as pores, but that allow things to diffuse across the membrane. However, it's worth noting they can be specific. So that's probably, you remember that little toy you had as a kid where you had sort of 3 different shaped objects, and then you had like a little there were 3 different holes, and one was circular, one was square, and one was triangular, and you had to sort of push the shape through the hole. And being the retards we were at the age of probably 1.5, we would always try and hammer the triangle shape through the through the circle hole. Not to much effect. Similar idea here. The shape of the tunnel, and also whether it's charged or simply polar, or the position of those charges can act like a keyhole and selectively let only certain molecules through. So the geometry, a lot like if you've done some stuff on enzymes, a lot like that complementary fit between an enzyme and its substrate. So think about, for instance, if this, if you had some sort of molecule, and if it had a positive region and a negative region to it, then the tunnel, the pore, could have sort of one side that has slightly negative charges and one that has slightly positive charges to accommodate that molecule through. So they can be quite specific. They can actually be quite specific tunnels to what's transported through them. But the process is entirely passive. It doesn't require energy. It's just simple diffusion from an area of higher concentration to an area of lower concentration. So there, for instance, sugars and amino acids are too big to fit through the plasma membrane, so they often enter through transport proteins. There's a little example for you. So here, lastly, facilitated diffusion is similar to osmosis and diffusion because they use a concentration gradient, and the key idea here is no energy is needed for that. The kinetic energy of the molecules is enough itself. So it is different from diffusion and osmosis because essentially this is a mechanism that allows molecules that are too big that are hydrophilic or polar or highly charged to essentially fit through or pass through the membrane. So especially really large molecules, are going to rely on some sort of protein to transport them across. So that's facilitated diffusion. We'll look at active transport in the next video.